So, good afternoon. We're beginning chapter 22. I have started going over your last exams, and I want to start thinking about the next exam, and that's chapter 22, 23, and 24, I believe. And we're starting with the lymphatic system and the immune system. And one quick way to think about this, of course, is the lymphatic system is a series of ductal structures that collect the interstitial fluid inside your body. And its job, of course, is to present it to your immune system. Okay, a population of cells that have both innate and adaptive immune responses. And what the purpose of the chapter is going to be is how do you contrast cell-mediated and antibody-mediated immunity. So what this means is we're going to have populations of cells. You're going to have to learn their responsibilities, most of which you have a general idea of from chapter 19. Now we need to figure out how do we actually activate them precisely? How do we ensure that, say, antibody-mediated immunity does not turn on the host, meaning make an immune response against the host, whereas cell-mediated effects should, generally speaking, be rather non-specific. And that's the way, hopefully, I'm going to have you think about it. Let's see how it goes, though. What are we thinking about? Well, we're thinking about disease resistance, because remember, you're nothing but a bag of salt water. That is a meal. And that means you actually have two major responses, one of which is going to be non-specific. Whoops, that's not what I want to do with that. Non-specific, which is, in this case, if we think about innate immunity. It's present at birth, okay, and provides general protection against the invasion of a wide range of pathogens. That is Babel speak for you have a series of white blood cells, natural killer cells, neutrophils, macrophages. Their job is to run around your body and go, are you friend, are you foe? And if you're a friend, we won't do anything to you. If you're a foe, we're going to kill you. Primarily, we're thinking about this in terms of, say, a bacterial response. Adaptive immunity, though, requires a specific white blood cell, a lymphocyte, to actually be activated by a unique pathogen, a particular pathogen, or a foreign substance that's detected inside the body. And these are, as you're going to expect, a multi-step set of activation systems that you're going to have to learn. And yes, it's difficult, but all we actually expect you to learn in this chapter is the general mechanism of activation. Beyond that, we're going to talk about other immune responses inside the lymphatic system in such a way that you really do walk out of this class understanding that you may have a passive immune system. Remember, the outside of your body is cutaneous tissue, and if all the bacteria on the body are trying to turn you into a meal, you have to remember that's both inside and outside. How does Mother Nature stop that from happening? Should something get into your body, which we know already happens, then what are the more adaptive responses? How does it know what to triage the energy towards to make sure it keeps you alive? Here in table 22.5 is a summary of the functions of cells participating in adaptive immune responses. And notice here, these are actually also macrophage and dendritic cells are non-specific cells, but what happens, of course, is they actually are responsible for activating the adaptive immune responses. And you'll see why this is really important. But you're going to notice that lymphocytes, remember we taught them to you in chapter 19, they came in a T cell variety and they came in a B cell variety and a natural killer cell. And you'll notice here we have many different responsibilities. So pay attention to that when we actually get to that part of the chapter. What is that lymphatic system? Well, as I told you a moment ago, it is the structures and organs that contain lymphatic tissue. Primarily, you're going to think of the thymus, the spleen, bone marrow. You might not necessarily think of bone marrow, but of course this is where we make all of our white blood cells, and the fluid that flows inside the lymph structure within the lymph vessel, which is collected interstitial fluid. Okay? From lab, you can recognize this cartoon, and primarily, remember, all fluid's going to flow primarily towards the cisterna chyli, where it's the lowest pressure point, draining from, say, Peyer's patches in your gastrointestinal tract and or the rest of your body. and will be deposited back into your venous blood return, like I stressed inside lab. What I want you to see here, of course, is not that you just have these small areas, but think about what Mother Nature is doing here. If you have an appendage, or a leg, or an arm, or even your neck, these have to drain into large blood vessels where the blood pressure is getting lower. The movement of these vessels, because the musculature is moving, is what affords the ability to the fluid to move back to the area of lower pressure. If you're pushing on these things, you're creating higher pressure. They're going to move towards lower pressure, and ultimately the respiratory pump of the thoracic cavity will move this material back into the circulatory system. Why is it doing that, of course, is, what you don't necessarily appreciate, but I know I've already mentioned to you, is that inside each of these lymph nodes, we're going to be surveying what's actually going on inside of there. Okay? So the lymphatic system is going to drain interstitial fluid. You're going to learn that it's also going to transport dietary fats and also carry out immune responses. If there's something that's been found inside the body that requires that immune response. So how does it actually do this? You've seen me draw something like this inside the class. And what you can see here, of course, is arterial blood flow traveling, creating a capillary bed, and excess fluid will be collected into those lymphatic structures. And now today we're going to add in a little bit more detail. And you can see here that if this is the open circulatory system of the lymphatic system, you have these anchoring filaments that are actually attached to cells in between the cells that are being fed by the capillary bed here. So they actually have purchase, is what we would call that, right? So the cells are holding all of this in place. The fluid, as it increases, let's go back here. As the fluid pressure increases, what actually happens, of course, is the fluid is now introduced into this. That interstitial fluid is now called lymph. Okay? So again, it's a pressure mechanism that actually starts all of this. And what's going to happen, of course, is the lymphatic vessels will pass through lymph nodes and then pass into lymph trunks. And the lymph trunks include the lumbar, intestinal, bronchomediastinal, subclavian, and jugular trunks, right? You definitely saw those inside lab, but here's an image of it in a little more detail where you can see here traveling up through the thoracic trunk itself, delivering itself into the thoracic or left lymphatic trunk. Meanwhile, you actually have the right bronchomediastinal as well as drainage from the actual cervical and cerebral portions, as well as the arms or the appendages traveling now into that right lymphatic duct. And of course, this is superior vena cava. So materials that have passed, stop and think about this for a minute. Materials that have passed through, let's go like this. Materials that have passed through a lymphatic structure, right? Let's see if I can get up there and do that. You're not behaving well. Maybe we won't be doing this today. So we've gone through the lymphatic structure. Okay, let's just do this. Gone through the lymphatic structures. They've passed through lymph nodes, which means they've been cleaned, which means, in theory, everything that's coming in these green arrows here has been surveyed materials. And what I mean by surveyed is they've passed through the cells that you actually find inside of a lymph node. And we'll come to the lymph node in a few minutes, but for right now, what I want to make sure you understand is by the time it gets back to either the right subclavian trunk, okay, or the left subclavian trunk, that material has been passed over the cells inside of the lymph nodes. A moment ago, I told you that the lymphatic organs and tissues, there are primary lymphatic organs where immune cells become immunocompetent. And what I mean by that is, imagine, if you will, Mother Nature ensures that red bone marrow is going to give rise to T cells and B cells. And these B cells and T cells have to get to a specific activation state. Then they're going to move someplace else. So they leave the bone marrow, and imagine T cells now travel to the thymus to actually mature. And what I mean by that is they're going to sit on the reticular branches And those, reticular's not spelled right there, but don't worry about it. Reticular branches, and they're going to mature. And then what they'll do is they'll migrate to the lymph nodes and or other structures, lymph nodes and or other structures inside the body, where they are actually looking for the molecule that will actually activate them. Reticular. Let's spell that right. It's driving me crazy. Okay? So that's what we mean by primary lymphatic structure. Here's what you already know where the thymus is. You know it is superior to the heart. I should say it's anterior to the heart. It's, I guess it is superior, anterior. It's deep to the sternum. You can see it's not to be confused with the thyroid gland. Okay? I know I joke about that, but really, you shouldn't be able to leave this class without even being able to distinguish those differences, right? And what it actually looks like inside is you actually have reticular tissue that makes up branches that are surrounded by a capsule that form a cortex that have within it a corpuscle, and this makes up the medulla. And inside of these, you can see the primary population of cells are T cells. These are all these little cells here. And you've seen them inside blood, so you have a general idea what they look like. You should be asking yourself, if they're inside the thymus, what are they doing inside the blood? Well, obviously, they're being transported. That's one of the responsibilities of the circulatory system, right? Secondary lymphatic organs and tissues include that lymph node, which you've seen inside lab. And remember, I've warned you, you have to be able to make sure you can describe the structure of this, S-T-R-U-T-U-R-E, of this tissue, because you need to be able to see that we're creating these sort of miniature pressure gradients that are going to allow for perfusion of lymphatic tissue over the surface of T cells and B cells. Spleen we'll come to in a few minutes. It has a slightly different responsibility, but essentially B cells and red blood cells, red BCs, are going to function and or at least be hematopoietically regulated here. And lymph nodes are, of course, similar to, but not necessarily identified as. Let's take a look at one of those lymph nodes here. We'll finish up this first segment by actually looking at a lymph node. And here's where things are different from lab. Notice what I'm doing when we're looking at the cortex here is we are looking at the cells that make up the outer germinal center of the cortex. So take a look at that. It's outside here versus, say, the inner cortex versus the inner germinal center of the outer cortex. And what I want you to pay attention to is you change the population of cells that are found inside the cortex of a lymph node. You have T cells and dendritic cells versus B cells, macrophages, and follicular dendritic cells. What we haven't told you yet is to activate T cells and B cells, you actually need to go up to them and say, hey, look what I found. And that's the purpose of dendritic cells. They act as what we're going to call antigen-presenting cells. So let me see if I can put that in there. Grab this stupid thing, see if I can get it to work now. Okay, we're going to go over here. It's completely backwards. This is not working well today. That's me, though. I'm just going to write A. No, I'm not going to write A. I don't know what I'm writing here, so let's stop that. Let's not even try and do that. Let's just put text box inside there. I'm going to put that right there. I'm going to go right there. I'm going to make sure you see APC. Okay? And that is antigen presenting cell. Antigen presenting cells. And what I want to make sure you understand here is that those follicular dendritic cells, those macrophages, dendritic cells, they have the ability to present materials on their surface so that the T cells and the B cells can know what's actually happening outside of the lymph node. And that's the really important idea here. As you move into the medulla, we change the population from B cells to plasma cells. Well, you remember what a plasma cell is. A plasma cell is an activated B cell. So something had to have happened between here and here. And what I need you to understand is every single plasma cell is unique. And you still have macrophages in here. They can clean out debris as cells begin to die or something is actually happening inside of them. And the flow of the fluid is this. Afferent flow of fluid into the subcapsular space perfuses over all of these cells till we travel to the medulla, where it then exits now through the efferent lymphatic vessels. So we're going to stop here. Okay, we'll pick up with the photomicrograph of the structure of the lymph node in a moment, okay, if you decide to watch it, and then we're going to transfer onto the spleen and see where things go from there. Okay, bye.